Hello, everyone. My pleasure to present a talk entitled The Fuzzy Entropy of Biomolecules and its Role for Life. The outline of my talk. First, I will present life as a remarkable emergent property of certain complex systems. Then I will show that molecules, especially macromolecules, are microscopic instances of complex systems. I will demonstrate that the distributions of conformational isomers of any compound is a molecular fuzzy set and a quantum mixed state. Its manipulation corresponds to processing fuzzy information, which is a form of quantum parallelism. Conformational isomers of biomacromolecules are fundamental for life. And finally, I will conclude by presenting some perspectives. Let's get started. Life is without any doubt the most staggering emergent property that certain complex systems manifest. From a chemical point of view, all living beings are made of lipids, DNA, RNA, proteins, water, salts, sugars, and so on. Taken separately, these compounds can never show life. Only when all those compounds are assembled in that peculiar structural and functional spatiotemporal organization that is a cell can life emerge. Life can be defined as a sequence of temporary events that starts with birth and continues with growth, usually reproduction, aging, and finally death. During these events, any living being manifests purposefulness through its intelligence, which is the ability to solve problems in a flexible and scalable manner. Cells can reach their fundamental goals by exploiting matter and energy to encode, collect, store, process, and communicate information. The basic information required for survival is stored in DNA, and the chemical behavior of the genome, including all the epigenetic events having proteins and RNA as co-protagonists, can be compared to that of extraordinary software that is capable of facing variegated conditions the cell may encounter during its lifetime, and rendering the self itself adaptable and evolvable. Cells are pristine examples of complex systems. But what are complex systems? Although numerous scientists have been investigating complex systems for many decades, as far as I know, one of the first seminal articles on complexity was Weaver's in 1948, and the label of complexity science became official in 1948 with the foundation of the American private research center Santa Fe Institute. We still lack a universally accepted definition of a complex systems. I like proposing an extensional definition of complex systems because it enumerates some systems that everybody would agree to include in the set of complex systems. If a single cell is the chemical prototype of a complex systems, then other examples surely are multicellular organisms, their societies, natural ecosystems, urban areas, the world economy, and the climate. However, I like adding that the set of complex systems is peculiar. The set of complex systems is a multivariable fuzzy set that includes numerous systems but at different degrees of membership. A fuzzy set is different from a classical set because it includes it breaks the law excluding middle. An element may belong to a fuzzy set with a certain degree, which can be any real number included between 0 and 1. The variables defining the complex system fuzzy sets are the number of elements, representing the multiplicity of the system, the diversity of the elements, the variability of the behavior of the elements over time, but also the number of links intertwining the elements, the so-called interconnection of the system, the diversity of the links, the variability of the links over time, and the extent of integration of the features, elements, and links giving rise to peculiar emergent properties of the whole system, the so-called integration. All these variables characterizing the elements and links, and especially the integration, depend on the context where the system is embedded. The degree of membership of systems to the multivariable complex system fuzzy sets grows by increasing the number of nodes, their diversity and variability, the number of links, their diversity and variability, and more importantly, the integration. Hence, the complexity of any system is a 7-variable function which is context dependent. This definition of system complexity depends on the level of description. If, for example, is at the molecular or cellular or organismic level, and includes both synchronic and diachronic complexity because it regards both the structural features of systems at one point in time, but also their time evolution. In general, complex systems are networks that operate out of thermodynamic equilibrium and exhibit emergent properties. But these are also the basic features of any molecule. For instance, any amino acid is a three-dimensional network whose nodes are atoms and whose links are bonds and interatomic electrostatic interactions operating over space. The molecule has features that are completely different from those of its constituent atoms. And these features are further enriched when the molecule is out of thermodynamic equilibrium. The structure of any molecule is fully known when the following features are specified. First, the type and abundance of every chemical element that constitutes the molecule, indicated through the molecular formula. The covalent bonds linking the different atoms, indicated through the structural formula. And the orientation of the atoms in the three-dimensional space, and their mutual electrostatic interactions operating through space. The structure of a molecule within atoms can be encoded in a square n/n adjacency matrix. The terms Aij of the matrix will be null if the two atoms I and J are not covalently bound and are far apart from each other in the three-dimensional space, so they do not even interact electrostatically appreciably. In all other circumstances, the terms of the adjacency matrix will be non-zero. Molecules that have identical molecular and structural formulas, but differ in the orientation of the atoms in the three-dimensional space, are named as stereoisomers. Among all the infinite possible number of stereoisomers, a finite collection of them correspond to minima of molecular potential energy. Considering the energetic barriers separating these minima, all the stereoisomers are grouped into two sets based on the energy required to interconvert between them. If this energy is of the order of a few electron volts or higher, enough to break covalent bonds, the stereoisomers are configurational. On the other hand, if the amount of energy is as large as or 10 times larger than the thermal energy available to every molecule at room temperature, which is of the order of 0.02 electron volt, then the stereoisomers are conformational, also called conformers. Conformers can interconvert by thermal energy and fluctuations. The rate of interconversion depends on the height of the energetic barriers. The higher the barrier, the slower the interconversion. When these barriers are as high as the available thermal energy, interconversion are ultra-fast, occurring in the time domain of hundreds of femtoseconds. Intramolecular steady hindrance from bulky molecular groups, intramolecular hydrogen bonds, viscous microenvironments, and low temperatures can hinder the rates of conformational interconversion, also called conformational dynamicity. Any compound that exists as an ensemble of NC conformational stereoisomers is described by a collection gamma of NC adjacency matrix of the type alpha, each multiplied by weight coefficient Wk, which represent the relative abundance of the K conformer. The sum of all the weights coefficient Wk is equal to 1. The authentic identity of any compound is appropriately represented by gamma, which is sensitive to the physicochemical features of its chemical context. These features are determined by specifying the qualitative and quantitative chemical compositions, including the solvent, the temperature, the pressure, the local electric and magnetic fields, and their dependence on the spatial and temporal coordinates. If the variables appearing in the definition of the physicochemical context assume uniform values in space, the chemical context is homogeneous, and any compound will exist as a unique gamma. On the other hand, when at least one of the variables appearing in the definition of the physicochemical context is not uniform in space, the microenvironment is heterogeneous. In any heterogeneous microenvironment, there will be more than one context, and hence more than one gamma, with as many gamma as are the distinct chemical contexts. Every living cell exhibits a high degree of microheterogeneity. Therefore, every biomolecule can be exposed to many chemical contexts and show a variety of conformational distributions and behaviors. The coefficient Wk appearing in the definition of gamma might depend on the history of the biomolecule they refer to. In other words, the final conformational distribution of a biomolecule might be affected by the pathways traced by the compound before reaching the final physicochemical context. This dependence gives rise to molecular level memory within a cell. Any conformational distribution of a compound is a molecular fuzzy set. Why? To answer this question, it's crucial to know the role that fuzzy sets play in fuzzy logic. Fuzzy logic reproduces the exclusive human ability to make decisions using deductive reasoning based on qualitative information expressed in natural language. Any nonlinear cause-and-effect relationship involving input and output variables is described through syllogistic statements of the type if-then, called fuzzy rules. And the fuzzy rules use adjectives for the input and output variables for graduating these variables. Adjectives are fuzzy sets that granulate and graduate the numerical values of both the input and output variables. For instance, in this figure, the variable temperature in the range -1 34 degrees Celsius is granulated and graduated through 4 fuzzy sets, and the number, position, shape, and graduation of the fuzzy sets are context-dependent features. The scheme reports the structure of any fuzzy logic system where we distinguish 3 elements: the fuzzifier that transforms numerical values of the input variables into degrees of membership to the input fuzzy sets, the fuzzy inference engine that transforms the activated input fuzzy sets into specific output fuzzy sets, and finally the defuzzifier that transforms the activated output fuzzy sets into a crisp value of the output variable exploited for a prediction or to make a decision related to the phenomenon described by the fuzzy logic system. One of the most iconic successes of fuzzy logic is its capacity to control the remarkably unstable inverted double pendulum. Since in certain circumstances, especially in the wild, living can be imagined as unstable as the state of the inverted double pendulum, my thesis that any living being can survive because it processes fuzzy logic at different spatial scales, from the molecular to the organismic level. In this contribution, I will focus on how fuzzy logic is processed at the molecular level. At the molecular level, when a chemical compound exists as a collection of distinct conformers and/or experiences microenvironments with distinct physicochemical features, it works as a molecular fuzzy set. This thesis relies on 2 observations. Such a compound existing in a conformational distribution will have context-dependent features and will granulate physicochemical variables. Now I give you a demonstration using this merocyanine, which is a pretty flexible structure and can exist in many conformers. The structures depicted in figure have the same molecular and structural formulas, but differ in the 3-dimensional orientation of their atoms. They are conformational isomers. A merocyanine is produced by UV radiation of a spiroxazine, which is direct thermally reversible photochromic compound. The structural change is accompanied by a significant spectral modification. The spiroxazine is uncolored, whereas the merocyanine also absorbs in the visible region, and when the absorbance in the visible region is recorded under UV radiation, it grows exponentially until a plateau is reached, indicating that the photochromic system has reached the photostationary state. As soon as UV radiation is stopped, the original equilibrium state is recovered because the merocyanine is a metastable species, and it transforms back to the original spiral form. Since the merocyanine can exist in many conformers, each having a peculiar lifetime, it's reasonable to use polyexponential function to fit the bleaching kinetics, and the best fitting function can be determined using the maximum entropy method, as its output is the least biased estimate possible from the experimental data. This figure shows some results for the merosonide dissolved in pure ethanol at different temperatures. The higher the temperature, the broader the distribution of the lifetimes, and the shorter the lifetimes. Lifetime distribution can be affected by chemicals. For example, by injecting glycine. Glycine exerts a double effect. It shrinks the distribution and lengthens the lifetimes. This double effect is presumably due to supramolecular interaction of glycine and the zwitterion uniforms of merosonide. Like this, the lifetime distribution is associated with a conformational distribution of merosonide. The shape and position of this conformational distribution depend on the physicochemical context, and the conformational distribution granulates a portion of the variable time. Therefore, it's evident that the conformational distribution of merosonide has the features of a molecular fuzzy set. Moreover, since each conformer is described by a peculiar wave function, the conformational distribution is also a quantum mixed state. In its definition, Wi prime represents the probability of the ith wave function, but also the degree of membership of the ith wave function to the molecular fuzzy set. The amount of information encoded by this conformational distribution is given by its fuzzy entropy, measuring fits. When molecular fuzzy sets are manipulated, fuzzy information is processed, which is an unconventional form of quantum parallelism. The data reported in the figure shows that the temperature decompresses the fits. It's a decoder. Fuzzy entropy changes from 0.24 at 292 Kelvin up to 0.74 at 308 Kelvin. On the other hand, glycine compresses fits. It's an encoder, and fuzzy entropy changes from 0.06 at 292 degree Kelvin up to 0.19 at 308 Kelvin. Conformation are relevant for molecules, but even more for macromolecules, as is evident for the biopolymers. Originally, it was thought that proteins existed in a specific unit 3D structure, and that structure was engineered to host a specific substrate. The unique complementarity between the rigid structure of a substrate and an enzyme was suggested to explain the efficiency of catalysis, and the lock and key paradigm was proposed. After collecting additional structural data on proteins, it's now evident that ordered proteins exist as dynamic ensembles of interconverting conformations, and many proteins are intrinsically disordered. Conformational heterogeneity confers multifunctionality to proteins. Each protein can exert multiple functions and exhibit functional promiscuity. Since the conformational distribution of any protein depends on the physicochemical context, proteins are adaptable and can even moonlight. Protein conformations are also crucial for the immune system, function of defending organisms against an almost infinite number of antigens. In every human diploid cells, there are about 2 meters of DNA that fit inside the nucleus of a cell, which is a few micrometers large because it's a fractal-like structure. DNA wraps around histone proteins to form nucleosomes. Nucleosomes coil into chromatin fibers, and chromatin fibers fold into a chromosome. The three-dimensional conformational organization of DNA is fundamental for the processes it participates in: gene transcription, regulation, and DNA replication. For instance, DNA conformations generated by bending, looping, and supercoiling affect transcription events by influencing the accessibility of genes to transcription machinery and regulatory proteins. RNA is an intrinsically highly flexible molecule, and in principle it can adopt many conformations adapting to its physicochemical context. RNA exhibits a structural and functional diversity comparable to that of intrinsically disordered proteins, including enzymatic activities. In summary, the reactivity of conformational distributions guarantees a significant computational parallelism. It's possible to increase the degree of parallelism by preparing mixtures of different quantum mixed states. which means by preparing mixtures of different molecular fuzzy sets. The chemical composition of these mixtures should be fixed by considering the granulation of the physicochemical variables. In this regard, a remarkable example is offered by human color vision. In the center of the retina, every human has three types of cones that have three distinct types of photoreceptor proteins, the so-called red, green, and blue proteins. These proteins share the same chromophore, which is the photochromic 11-cis retinal. However, the three types of proteins differ in the spectral position of the lowest energy absorption band, because they differ in the amino acid composition of the pocket embedding the retina. The three characteristic bands are associated with distinct transition moment integrals. They are characterized by peculiar vibronic wave functions. The three absorption bands are three molecular fuzzy sets, granulated in the visible region, and lights with distinct spectral compositions belong to these three molecular fuzzy sets to varying degrees. The collection of different degrees is interpreted as a peculiar hue in our brain. The granulation of the visible region is a form of quantum parallelism that enables humans to distinguish around 200 hues. Starting from the premise that the spectral granulation of the visible region allows humans to distinguish colors, knowing that direct thermally reversible photochromic compounds absorb UV and become color, is it possible to granulate the UV through a photochromic system capable of originating different hues of colors depending on the UV regions, UVA, UVB, and UVC? The answer is that one compound is not enough, because the position of the band generated in the visible region by one photochromic compound is always the same, whatever the excitatory wavelength in the UV. On the other hand, it has been demonstrated to be feasible by preparing a mixture of photochromic compounds. However, the composition of the mixture should be fixed, considering that the absorption bands of the uncolored forms should granulate the three UV regions, whereas the absorption bands of the colored forms should granulate the visible region. This slide reports the construction of a photochromic system based on four photochromic compounds. The absorption bands of the uncolored forms act as the fuzzifier, the photochromic reactions serve as the fuzzy inference engine, and the absorption bands of the colored forms are the output fuzzy sets. Finally, the human eye performs the defuzzification, and when the photochromic mixture is irradiated with UVA, UVB, or UVC wavelengths, the solution appears green, gray, or orange, respectively. It's evident that the biologically inspired photochromic fuzzy logic system extend human vision into the UV spectrum. In conclusions, any single cell is a parallel distributed processing molecular network having DNA, RNA, and proteins as its principal nodes. Given the relevance of conformational states in DNA, RNA, and proteins, it follows that all modules of the parallel distributed processing network that constitute a living cells are intrinsically fuzzy. Their fuzziness ensures adaptability and the ability to make decisions in environments characterized by uncertainty and vagueness. Any chemical reaction corresponds to the processing of fuzzy information, which is a form of quantum parallelism, since any molecular fuzzy set is also describable as a quantum mixed state. The fuzziness of biomolecules broadens the scope of the burgeoning field of quantum biology. Living organisms enhance their biological functions, not only by exploiting quantum coherent phenomena such as superposition, interference, tunneling, and entanglement, but also the computational parallelism of quantum mixed states that implement fuzzy sets. This charming property of life is inspirational for the research field of chemical AI, which mimics biological intelligence competencies using molecules and macromolecules as the hardware, and the chemical reactions, chemical waves, diffusion, migration, convection, and advection as the software of chemical systems maintained in thermodynamic out of equilibrium conditions and in liquid phase, that is the phase of life. I thank all of you for your attentions. I want to acknowledge my current collaborators, my past and present students, the European Union and my university for the financial support, and all of you for your attention. You can follow me on the social media because I like disseminating the subject of complexity and chemical AI. Thank you for your attention.